we come across several varieties of plants every day. The internal structures of these plants have some similarities and some differences. The study of the internal structure of plants is known as anatomy. Let's understand the anatomy of a flowering plant. A plant is formed of different types of tissues. Tissue refers to a group of cells that have a similar structure and perform the same function. There are two main types of plant tissues: meristematic tissues and permanent tissues. Meristematic tissues undergo active cell division, while the permanent tissues do not undergo any division. The word meristem has been derived from the Greek word meris, which means division. The specialized region in plants where meristematic tissues are present is known as the meristem. Active cell division takes place in this region, leading to the growth of the plant. After meristematic division, the newly formed cells become structurally and functionally specialized and lose the ability to divide. These cells are also known as permanent cells or mature cells. There are two basic types of meristems: primary and secondary. Primary meristems help in the development of the primary plant body. They are present throughout the life of the plant. Primary meristem can be further divided into two types: apical meristem and intercalary meristem. Apical meristems are named depending on their location in the plant's body. The meristem found at the tips of the roots is known as root apical meristem, whereas the meristem found at the tip of the shoot is known as shoot apical meristem. During the formation of the leaves and the elongation of the stem, some cells are left behind from the shoot apical meristem. These cells form axillary buds. Axillary buds are formed at the axil of the leaves and can develop into a branch or a flower. During the formation of the primary plant body, some particular regions of the apical meristem produce dermal tissues, ground tissues, and vascular tissues. Another type of primary meristem is intercalary meristem, which occurs between mature tissues. It helps in regenerating grass blades removed or eaten by grazing animals. Meristem that appears later in a plant's life is called secondary or lateral meristem. It occurs in the mature regions of the roots and shoots of many plants. Secondary meristem is responsible for producing woody axis and secondary tissues. Fascicular vascular cambium, interfascicular cambium, and cork cambium are some examples of secondary meristems. Thus, meristematic tissues play a vital role in the primary and secondary growth of a plant. Like all living organisms, the cells in a plant are also grouped into various tissues. Permanent plant tissue is a group of cells which are complete in growth and usually incapable of meristematic activity. Permanent plant tissue is of two types: simple permanent tissues and complex permanent tissues. Complex permanent tissues are a group of more than one type of cells that have a common origin and work together as a unit. There are two types of complex permanent tissues in plants: xylem and phloem. Xylem performs the function of conduction of water and minerals from the roots to the stem and leaves. It also provides mechanical strength to the plant. Xylem consists of 4 different types of cells: tracheids, vessels, xylem fibers, and xylem parenchyma. Tracheids are elongated, angular, lignified cells with tapering ends. They are dead and without any proptoplasm. The inner layers of the cell walls in tracheids vary in thickness. Vessels are long, tube-like structures composed of many cells called vessel members. Vessel members have lignified walls and a large central cavity. They are interconnected through perforations in their common walls. Typically, vessels are found in all angiosperms, and vessel cells do not have protoplasm. Xylem fibers are sclerenchymatous fibers associated with the xylem. They have highly thickened walls and obliterated central lumens. These may be septate or aseptate. Xylem parenchyma is composed of parenchymatous cells. They are living and thin-walled, with cell walls made up of cellulose. These cells store food material as starch or fat, or substances such as tannin. Some xylem cells are arranged in the form of a ray through which radial conduction of water takes place. These cells are known as xylem ray parenchymatous cells. They are produced from the differentiation of vascular cambium. We'll now learn about primary xylem, which consists of the young cells produced from meristematic tissues by the differentiation process. Primary xylem is of 2 types: protoxylem and metaxylem. The primary xylem elements formed in the beginning of a plant's life are known as protoxylem. On the other hand, the primary xylem elements formed in the later stages are known as metaxylem. The arrangements of primary xylem are end-arc and exarch. In the stem, the protoxylem lies towards the center or pith, and the metaxylem lies towards the periphery of the stem. This arrangement of primary xylem is called end-arc. Let's talk about the roots now. Here, the protoxylem lies towards the periphery and the metaxylem lies towards the center. This arrangement of primary xylem is known as exarch. The second complex permanent tissue that transports food materials from the leaves to the other parts is known as phloem. In angiosperms, the phloem consists of sieve tube elements, companion cells, phloem parenchyma, and phloem fibers. Sieve tube elements are tube-like structures that are arranged in a longitudinal manner. They are closely associated with companion cells, which are parenchymatous in nature. The end walls of sieve tube elements are perforated in a sieve-like manner and form sieve plates. The functions of sieve tubes are controlled by the nucleus of the companion cells. The sieve tube elements and companion cells are connected by pit fields present between their common longitudinal walls. The companion cells also help in maintaining the pressure gradient in the sieve tubes. phloem parenchyma stores food material and other substances like resins, latex, and mucilage. It consists of elongated, tapering cylindrical cells that have dense cytoplasm and nucleus. The cell wall of phloem parenchyma is made up of cellulose. It has pits through which plasmodesmatal connections exist between the cells. Phloem fibers or bast fibers are made up of sclerenchymatous cells found in secondary phloem. They are elongated, unbranched with pointed apices, and their cell wall is quite thick. On maturity, phloem fibers lose their protoplasm and become dead. Jute, flax, and hemp are some examples of commercially used phloem fibers. Complex permanent tissues are engaged in the transportation of water, minerals, nutrients, and organic substances. Therefore, they are extremely essential for the survival of plants. Permanent plant tissue is a group of cells which are complete in growth and usually incapable of meristematic activity. Permanent tissue is of two types: simple permanent tissues, where all cells are similar in structure and function, and complex permanent tissues, which are composed of different types of cells. Let's learn more about simple tissues. There are three types of simple permanent tissues: parenchyma, cholenchyma, and sclerenchyma. Parenchyma is the most common ground tissue. It performs functions such as photosynthesis, storage, and secretion. The cells of the parenchyma have the same dimensions or the same length, width, and height, which means that they are isodiametric. Parenchymatous cells are of different shapes, such as sphere, oval, round, polygonal, or elongated. The cell walls are thin and made up of cellulose. Parenchymatic cells may be closely packed or have small intercellular spaces between them. The second type of simple permanent tissue is cholenchyma. It is found in the layers below the epidermis of dicotyledonous plants. This layer is either homogeneous or in patches. Cholenchyma provides mechanical support to the growing parts of a plant, such as a young stem or the petiole of a leaf. The cells of the cholenchyma are thick at the corners due to cellulose, hemicellulose, and pectin deposits. They may be oval, spherical, or polygonal in shape, and there is no intercellular space between them. These cells often contain chloroplasts and assimilate food by the photosynthetic processes. The third type of simple permanent tissue is sclerenchyma, which consists of long, narrow cells with thick lignified cell walls. In fact, the term sclerenchyma has been derived from the Greek word skleros, which means hard. Sclerenchyma provides stiffness or mechanical support to plant organs. The cell walls of sclerenchyma have a few or many pits. These cells are generally dead and without any protoplasts. Based on the variation in form, structure, origin, and development, sclerenchyma tissue can be of two types: fibers and sclereids. Fibers are narrow, elongated, spindle-shaped, thick-walled cells with pointed ends. They usually occur in groups in various parts of the plant. Sclereids are short and thick-walled cells, generally spherical, oval, or cylindrical in shape. They are extremely lignified and have narrow cavities or lumen. They occur in the hard covering of seeds, the shells of nuts, and the pulp of fruits like guava, pear, and sapota. Simple permanent tissues are very essential as they provide protection, storage, support, and strength to the plant. The body of a plant consists of various types of cells which form different types of tissues. The structure and function of these tissues depend entirely on where they are located inside a plant. Therefore, based on this, plant tissue systems can be classified into three types: epidermal tissue system, ground or fundamental tissue system, and vascular or conducting tissue system. Let's begin by studying the epidermal tissue system. As the name suggests, this tissue forms the outermost covering of the plant body. It consists of epidermal cells, stomata, and epidermal appendages, including trichomes and hairs. Epidermis refers to the outermost layer of the primary plant body. It is a continuous layer of elongated and compactly arranged cells. Epidermal cells are parenchymatous in nature, and their cell walls are lined with a small amount of cytoplasm that has a large vacuole. The outer layer of the epidermis is coated with a thick and waxy layer called the cuticle, which prevents the loss of water. Stomata are present in the epidermis of the leaves. They regulate the process of transpiration and gaseous exchange. Each stoma consists of two bean-shaped cells called guard cells. Guard cells contain chloroplasts and regulate the opening and closing of a stoma. In grasses, the guard cells are dumbbell-shaped. The outer walls of the guard cells, which are away from the stomatal pore, are thin, while the inner walls, which are towards the stomatal pore, are very thick. When the epidermal cells associated with the guard cells become specialized in shape and size, they are known as subsidiary cells. The stomatal aperture, guard cells, and the surrounding subsidiary cells are collectively called stomatal apparatus. The epidermal tissue has epidermal appendages such as hair and trichomes. The root hairs are unicellular elongations of the epidermal cells, and they help absorb water and minerals from the soil. The epidermal hairs on the stem are known as trichomes. They may be branched, unbranched, soft or stiff, and may also be secretory in function. Besides this, trichomes prevent loss of water due to transpiration. The second type of tissue system is the ground tissue system. the ground tissue in primary stems and roots, consists of parenchymatous cells found in the cortex, pericycle, pith, and medullary rays. In the leaves, ground tissue is composed of thin-walled chloroplast-containing cells, collectively called mesophyll. Let's now study the third type of tissue system, the vascular tissue system. It is composed of complex tissues, namely xylem and phloem, which together form vascular bundles. In the vascular bundles of dicotyledonous plants, cambium is present between the phloem and xylem. Due to the presence of cambium, such vascular bundles can form secondary xylem and phloem tissues and are known as open vascular bundles. On the other hand, in monocotyledonous plants, vascular bundles do not contain cambium. They are unable to form secondary xylem and phloem tissues and are thus called closed vascular bundles. There are two types of vascular bundle arrangements, radial and conjoint. The radial arrangement is generally found in roots, where the xylem and phloem in a vascular bundle are arranged in an alternate manner at different radii. In the conjoint arrangement found in stem and leaves, the xylem and phloem are situated at the same radius of the vascular bundles. The conjoint vascular bundles usually have the phloem located only on the outer side of the xylem. In the conjoint open arrangement, the cambium is present between the xylem and phloem, while in the conjoint closed arrangement, the cambium is absent. The plant body has an intricate arrangement of various tissue systems that aid its growth and development. The root is one of the key organs that helps distinguish between a dicot and a monocot plant. Let's observe the transverse section of the matured zone of roots to understand their internal structure. We'll begin by studying the anatomy of a dicot root. The outermost single-layered tissue of the dicot root is called the epidermis. On the epidermis, you will also find some extended epidermal cells, which are unicellular root hairs. The epidermis is followed by cortex, which is formed of several layers of thin-walled parenchymatous cells. These cells have intercellular spaces. The innermost layer of the cortex comprises barrel-shaped cells without any intercellular spaces. This layer is known as the endodermis. The tangential and radial walls of the endodermal cells have deposits of a water-impermeable material or suberin, which is also called Casparian strips. Suberin is a waxy, waterproof substance present in the cell walls of the cork tissue in plants. Going back to the transverse section of the dicot root, after the endodermis, a few layers of thick-walled parenchymatous cells are present in the tissue. This is known as the pericycle. Secondary growth, such as initiation of lateral roots and vascular cambium, takes place in the cells of the pericycle. Notice that the pith in the tissue of the dicot root is extremely small and undeveloped. Typically, there are 2 to 4 xylem and phloem patches in a dicot root. In the later stages, a cambium ring gets formed between the xylem and the phloem. Some radially arranged parenchymatous cells are found between the vascular bundles. These cells form the conjunctive tissue, which is specialized for storage of water. The tissues present inside the endodermis, namely the pericycle, vascular bundles, and pith, are collectively known as stele. Let's now take a look at how the monocot root is organized internally. In many aspects, it is similar to the dicot root. On comparing the two roots, we find the monocot root also consists of epidermis, cortex, endodermis, pericycle, vascular bundle, and pith. However, there are some differences. The monocot root can have many xylem bundles, whereas the dicot root does not have quite as many. Moreover, in a monocot root, the pith is well developed. Although dicot and monocot roots have some structural similarities, they differ in the number of xylem bundles and size of the pith and cortex. Moreover, secondary growth takes place only in the dicot root. In other words, these roots thicken over time. A stem is the main axis or stalk of a plant, and it develops from the plumule of a germinating seed. Let's examine the internal structure of a stem in dicot and monocot plants. Here is the transverse section of a dicot stem. The regions of tissues in this stem include epidermis, cortex, and stele. The outermost layer of the stem, or epidermis, consists of a layer of closely packed cells and is covered with a thin waterproof layer called the cuticle. The epidermis also contains stomata and hair-like outgrowths known as trichomes. The next region of a dicot stem is the cortex. It lies between the epidermis and the pericycle and consists of multiple layers of cells. The main function of this region is storage of food material. The cortex comprises three subzones, namely the hypodermis, the cortical layers, and the endodermis. The hypodermis is the outermost zone of the cortex, which consists of a few layers of parenchymatous cells. These cells strengthen the young stem of a plant. Below the hypodermis are cortical layers of round and thin-walled parenchymatous cells with intercellular spaces. The third or the innermost zone of the cortex is known as the endodermis. It is a single layer of tightly packed rectangular cells which are rich in starch grains. Therefore, this layer is also known as the starch sheath. Let's now examine the composition of the stele, which forms the last region of a dicot stem. This region consists of the pericycle, the vascular bundles, and the pith. The pericycle is composed of sclerenchymatous cells, and it lies on the inner side of the endodermis above the phloem cells. These cells act as a strengthening material. The vascular bundles are located in a ring on the inside of the pericycle. This ring arrangement is a distinctive feature of the dicot stem. There are layers of radially arranged parenchymatous cells in between the vascular bundles, which constitute the medullary ray. Each vascular bundle in a dicot stem is conjoint, open, and consists of end-arc protoxylem. The central region of the stem is filled with pith. It is composed of a large number of rounded parenchymatous cells with many intercellular spaces. Now that you've learnt about the dicot stem, let's study the monocot stem. The tissues of dicot and monocot stems are quite similar. However, there are some differences as well. In the monocot stem, the hypodermis is composed of sclerenchymatous cells, while in a dicot stem, it is composed of collenchymatous cells. The monocot stem has scattered vascular bundles and their arrangement is conjoint and closed. Moreover, water-containing cavities are present within the vascular bundles. On the other hand, in a dicot stem, the vascular bundles are arranged in a ring as conjoint and open. In a monocot stem, each vascular bundle is surrounded by a sclerenchymatous bundle sheath and a parenchymatous ground tissue. Moreover, the vascular bundles on the periphery are smaller compared to the vascular bundles in the center. Therefore, the essential differences between a monocot and dicot stem lies in the arrangement of the epidermis, ground tissue, and vascular tissue. Leaves are the most visible organs of a plant. They perform many crucial functions such as photosynthesis and transpiration, in which their internal structure plays an important role. A dicot leaf is also known as dorsiventral, as its upper and lower sides are different in structure. On observing the transverse section of a dicot leaf, we find that it has three main parts: epidermis, mesophyll, and the vascular system. The epidermis covers the upper and lower surfaces of the leaf and is coated with the cuticle. The epidermis covering the upper surface of leaf is called the adaxial epidermis, while that covering the lower surface is called the abaxial epidermis. Typically, the abaxial epidermis has more stomata compared to the adaxial epidermis, which sometimes may not have any stomata at all. The next part of the leaf is the mesophyll, which is located between the upper and the lower epidermis. It is also called the green tissue of the leaf, as it contains chloroplasts. The mesophyll is made up of parenchyma and performs photosynthesis. In dicot leaves, the mesophyll tissue is differentiated as palisade parenchyma and spongy parenchyma. The palisade parenchyma, located near the adaxial epidermis, is made up of elongated cells arranged vertically and parallel to each other. These cells have fewer intercellular spaces. The spongy parenchyma is located just below the palisade parenchyma, and it extends up to the abaxial epidermis. It consists of round cells which are loosely arranged and have large intercellular spaces and air cavities. The third part of a dicot leaf is the vascular system comprising vascular bundles. When observed under a microscope, vascular bundles can be seen in the veins and the midrib of the leaf. The size of the vascular bundles depends on the size of the vein. In dicot plants, the leaves have reticulate venation, in which the veins are of different sizes. Therefore, the vascular bundles are found irregularly scattered in the mesophyll. In the large veins, vascular bundles are usually surrounded by a bundle sheath. Let's take a look at a monocot leaf now. It is also known as an isobilateral leaf, which means it is identical on both sides of its axis and shows parallel venation. The anatomy of a monocot leaf is similar to that of a dicot leaf. However, there are also some key differences. Unlike a dicot leaf, where stomata are found in greater numbers in the abaxial epidermis, in a monocot leaf, stomata are present on both surfaces of the epidermis. Moreover, the mesophyll cells are not differentiated as palisade and spongy parenchyma in a monocot leaf. The vascular bundles in a dicot leaf are irregularly scattered due to reticulate venation. In a monocot leaf, however, they are arranged in a parallel manner in the mesophyll due to parallel venation. In the leaves of monocots such as grasses, veins and cells of the upper epidermis modify themselves as large, bubble-shaped, colorless cells, also known as bulliform cells. On absorption of water, bulliform cells become turgid and expose the leaf's surface. Conversely, in water stress situations, there is loss of turgor pressure and bulliform cells become flaccid. As a result, the leaves curl inwards to minimize water loss. Hence, a lot of activity goes on in the internal structure of a leaf. Since major food production takes place in leaves, they are aptly known as the plant's powerhouse of energy. The growth of plants is an ongoing process. Primary growth, which takes place due to the primary meristem, results in the lengthening of stems and roots. Another growth process begins after the period of primary growth and leads to the formation of the woody axis or an increase in the girth of the stem and roots. This is known as secondary growth. In the stem, secondary growth takes place due to the activity of secondary meristems such as the vascular cambium and cork cambium. The vascular cambium, located between the primary xylem and primary phloem of a vascular bundle, is also known as the intrafascicular cambium. When the stem is young, the vascular cambium is present as a patchy single layer. However, in the later stages, it develops into a complete ring, also known as the cambial ring. Let's understand how this ring is formed. When the secondary growth of a stem begins, a process of differentiation takes place in the cells of the medullary rays. These cells, located next to the intrafascicular cambium, become meristematic in nature and lead to the formation of the interfascicular cambium. Thus, the fusion of intrafascicular cambium and the interfascicular cambium results in the formation of a continuous ring of cambium or the cambial ring. At this stage, the cambial ring has primary xylem on its inner surface and primary phloem on its outer surface. Cell division takes place on both the sides of the cambial ring and results in the formation of secondary xylem on the inner surface and secondary phloem on the outer surface. Since the cambium is more active on the inner side, the formation of secondary xylem is more than secondary phloem. Due to the continued formation of secondary xylem, primary and secondary phloem get crushed. However, primary xylem remains intact in the center. The cambium also forms a narrow band of parenchyma at some places. This band passes through the secondary xylem and the secondary phloem in radial directions and is called secondary medullary rays. The secondary xylem tissues formed during cambial activity give rise to commercial wood. This cambium activity is impacted by several physiological and environmental factors such as defoliation, light, diseases, drought, and temperature. During some seasons, the cambium divides actively, while during other seasons its activity slows down. Consider the example of the temperate regions, where climatic conditions are not uniform throughout the year. During spring, when the cambium is active, it produces a lot of xylary elements with vessels that have wider cavities. Wood formed during this season is called spring wood or early wood. On the other hand, during winter, the cambium is less active. Hence, it forms fewer xylary elements with narrow vessels. Wood formed during autumn is called autumn wood or late wood. Spring wood is lighter in color and has a lower density, whereas autumn wood is darker with a higher density. These two types of wood appear as two concentric circles on the trunk and together represent the annual ring. In the central layers of the stem of an old tree, the color of secondary xylem turns dark brown due to the deposition of organic compounds such as tannins, resins, gums, and essential oils. These deposits make the wood hard and durable and resistant to microorganisms and insects. This region of the wood also consists of dead elements with highly lignified walls. It is called heartwood and provides mechanical support to the plant but does not conduct water. The peripheral region of the secondary xylem, which is lighter in color and is involved in conduction of water and minerals, is called sapwood. The cortex is another region where secondary growth takes place. Due to the vascular cambium's activity, the stem increases in girth. This results in rupturing of the cortical and epidermal layers. These layers need to be replaced with new protective cell layers. In due course, another meristematic tissue called cork cambium or phelogen develops, usually in the cortex region. In the phelogen, differentiation of cells begins on both sides. As a result, the cells of the inner surface differentiate into the secondary cortex or pheloderm, while the cells of the outer surface differentiate into cork or phellum. Tissue resulting from secondary growth in the cortex, namely cork, phellogen and phelloderm, are collectively called periderm. Tissues outside the vascular cambium, which includes the secondary phloem and periderm, are typically known as bark. Bark forms the outermost layer of the stem and roots of woody plants. Bark formed in the early season is called early or soft bark, while bark formed at the end of the season is called late or hard bark. Due to the formation of bark, gaseous exchange between the internal living cells and the outer atmosphere is cut off. To facilitate gaseous exchange, phellogen cuts off parenchymatous cells that break the epidermis and form lens-shaped openings on the stem called lenticels. Secondary growth in a dicot root is similar to that of a dicot stem. The vascular cambium develops from the pericycle tissue located just below the phloem bundles. Initially, the cambial ring is continuous and wavy, which later grows and becomes circular. Secondary growth of stems and roots occurs mainly in dicots and gymnosperms, while monocots lack secondary growth.